sugars, right? Carbs for short. And carbohydrates have been kind of vilified as a macromolecule lately. I remember late 80s and early to mid 90s, everything was fat-free, right? So there was fat-free cookies, fat-free chips, fat-free this. And the way to make them still taste okay was just to dump in a bunch of sugar, right? Because that was what was on everyone's radar, was fat, okay? And then they realized, well, man, it's got no fat, but it has a ton of calories in it. And, in the 90s, the diabetes epidemic really started to gain a lot of traction. And so it kind of switched from, okay, fat's okay, we're gonna do the Atkins diet where you can eat a bunch of fat, but no sugar. And, I feel like now it's just depending on who you ask, nothing but meat, nothing but plants, no sugar. Sugar's fine. So I'm not a nutritionist, and so I'm not going to give any advice on what I think. But from a biochemist perspective, we have to have sugar, right? We have to have sugar to make ATP. And ATP is what drives everything, right? Everyone knows what ATP is? Okay, yeah. Made in the mitochondria. It's done through glycolysis, breaking down glucose. Again, glucose is the preferred molecule, but fructose is also used, and some other sugars like maltose. But without sugar, we would cease to exist, right? So carbohydrates need to be consumed. However, we have only lived the last 100 years at the most having access to the sugar that we have now. And I would say even probably the last 20 to 30 years hasn't been, where it is in this day and age, where sugar is literally just added to everything. It's hard to find stuff that it doesn't have. And as a consequence, diabetes is one of the most significant contributors to mortality and morbidity in the US and other developed countries, right? And that's because our body has evolved to regulate glucose, right? So we have insulin. When our blood sugar concentration gets to a certain threshold, the insulin starts being produced by our pancreas and says, okay, let's shuttle this sugar into the cells so that they can start making ATP. Oh, and there's some left over, let's make that into glycogen. Glycogen is a polysaccharide, right? It's how we store leftover glucose. And then when that blood sugar balances out, then you have homeostasis. Once it starts to drop a little, and I think normal range is about 60 to 100 milligrams per deciliter. So when it drops below that, then the pancreas makes a bunch of glucagon, which then says, okay, let's pull that sugar, or that glycogen, let's pull those glycogen stores out of the liver and start breaking them down to glucose and use that. But imagine, back in our ancestral times, it's summertime out, it's night. You live in Tahoe, right? Where are you going to get glucose? It's not, I guess there's some berries out there, right? But it's hard, right? And in the wintertime, it's even harder, right? So we go through these periods where we have access to sugar and we use it, and then we store whatever we don't have in the form of adipose glucagon. And then there's this process called, you guys familiar with gluconeogenesis? It's essentially making glucose from non-carbohydrate sources. So when you are out of glucose, because, again, during evolutionary pressures that were on our, not just us, but mammals, right? In times where glucose wasn't readily available, you could get it from other things. You could get it from lipids. You can even convert things like alanine to pyruvate, which is what then goes on to phosphorylate ADP into ATP. Okay. None of this stuff is going to be relevant for the final, but just giving you kind of an introduction to what I think is important when we think about carbohydrates, because again, colloquially, carbs are have a certain connotation to them. But now we've gone from having scarcity to surplus, right? And you actually have to be really careful about what you consume, because what happens is if you have too much sugar in your blood, there's only so much insulin you can produce, right? And then you become desensitized. Your insulin sensitivity goes down. And there are two types of diabetes, right? Anybody know the difference between those two? There's 1 and type 2.
Yeah, type 1, 2.
Right? You know what type 1 is?
Isn't it something with the receptors? I don't know which one is which, but it's the receptors for insulin, and then one just doesn't make enough insulin, right?
Yeah, pretty much. So type 1 is an insulin insufficiency.
Okay.
And that's usually a genetic trait. So it's not acquired. Your body, just for whatever reason, doesn't produce enough insulin. So you do the injections, and that gives you normal endogenous. You're getting it exogenously, but it would give you the normal levels of what people without diabetes would have endogenously. Type 2 is insulin resistance, and that's where the receptors are that you're talking about. So that is usually acquired. It's usually acquired from a lifetime of certain behaviors where you're consuming so much sugar that your body just can't produce enough insulin to sequester it. You have no place to put it, so the insulin just stops doing its job. And then you have sugar coursing through your blood, which then starts to glycosylate your organs. When it's in your blood, it affects your circulatory system, right? So advanced stages of diabetes can, if you've seen people's limbs, they're often really purplish and red and swollen. It can lead to amputations, infections. Your immune system is plummeting because you are literally just full of sugar. Diabetes, mellitus, means pretty much like sweet pea, sugar urine. That's what it translates to. And interestingly, they used to, before we had tests to look at sugar, they used to taste it, because if you have so much sugar in your system that you can't process it, eventually it goes into your kidneys, and then it's excreted. So it will literally make your urine sugary. So now we have more hygienic ways of testing. You can do things like a Benedict's test to look for reducing sugars, which we'll talk about a little bit today. But so carbohydrates are important. We're going to look at it less from a biochemistry perspective, more of the organic chemistry perspective. But that's how it ties in, in my area of concern, right? Biomedically, it's a really important molecule, but too much of anything can be a bad thing. And that's what we're really facing now. All right, so a lot of things. I wanted to review that question that we left off with, or we bypassed on Tuesday, looking at nitriles and amides to synthesize amines. And then we can get into how we classify sugars it depends on what kind of carbonyl group they have. It's going to be either an aldehyde or a ketone. And then we can further determine if it's a D or L monosaccharide based on where a particular hydroxyl group is located. And then we'll talk about how these open chain monosaccharides close into a ring, or the cyclization. Get into just a few reactions. We're not going to cover all the reactions. Particularly, there are some reactions to make sugar chains longer or make them shorter. I'm not going to get into those, okay? But you could take a 4-carbon sugar and turn it into a 5-carbon sugar, or vice versa with certain reactions. And then get into disaccharides, lactose being one, like you mentioned, and then polysaccharides, glycogen. Okay. But first, let's review. So we had talked about on Tuesday how we can take an alkyl halide and just essentially do a substitution, right? With our sodium cyanide, put a nitrile group on there, and then reduce that with lithium aluminum hydride into just a regular amine, right? And that's going to be doable if we're just essentially tacking an amine on to the end of something. Okay. If we're going to attach an amine like in the middle, then this carboxylic acid is going to work better because we have this R group here, and we can attach it there and have it kind of sandwiched in between 2 R groups. But similar kind of thing, we use this thionyl chloride and then reduce it with lithium aluminum hydride again to get our amine. So I believe the question, is this the question that we did not do in class? And I said, let's work on that. Okay. Did anybody work on this?
Yeah.
Yeah, okay, great. So this first one, first of all, what would we call that? What would we call A? Going back to the nomenclature. What's that?
It's a propylamide?
Yeah, propanamide.
Propanamide.
Or propanamine.
Propanamine.
Right? And so, what could we use? We could use either a propane nitrile or a propane-based carboxylic acid. Right? So, was that propanoic acid?
Mm-hmm.
All right. And then using this propanamide would make our, so if we had, let's see, we had 1, 2, 3, and then is that right?
There's one too many carbons.
We just
So,
take,
yeah.
we leave that. There we go. I can do that. Right? And then we just reduce that by tacking on 1, 2. Now, the other option is to use propanamide, right? So that would be 1, 2, 3, and then we have NH2 coming off there, and a carboxyl group. carbonyl group, right? And then if we reduce that, 1, 2, 3. Okay. Cool. And then what about the next one? Where's the erase? Oh, there's hiding. Here, what is that molecule? So that has 2 propyl groups. So that, and how many hydrogens are here? Just 1. So when you see that, that indicates that this nitrogen must be sandwiched in the middle, right? So 1, 2, 3. 1, 2, 3. That's what we're going for, right? And so again, so this is a situation where we're not just tacking it on the end, so we couldn't use a nitrile, because a nitrile, right, has this. And so we can only attach it to that carbon, and then this is at the end. So if we use, what could we use?
You could have like, if like one of the propyls had like a nitrile on the end and the other one had a halogen, it could substitute maybe with that other one.
Okay, so we do use, you're saying we could use a nitrile.
On one of them, yeah, and then do like an SN2 with a halide on another propyl
And
group
switch
maybe.
them together.
Maybe. Kind of what I was thinking.
Problem with the cyanide is it's just 1 carbon, right? So we could attach this onto something, but how are we going to get something on this side?
On that side, okay.
What if we used this? What would we call this? So we would call this N-propyl propanamine,
Okay.
right? So you find one of whichever chain is longest. In this case, they're both the same, right? 1, 2, 3, 1, 2, 3. So it's a propanamine. And then we have another propyl group coming off of this, so we call it N-propyl propanamine. If we use the N-propyl propanamide, then we could just reduce that, right? So 1, 2, 3. That's, and then 2, 3. What would N-propyl propanamide look like? First, let's do an amide. This would be 1, 2, 3, 4. Okay, so there. 1, 2, 3. So now we have propanamide, right? N-propyl means 1, 2, 3. Then we have a propyl group there. So that's going to be using this, and this R, right, is a 1, 2, 3. Just draw it backwards, and then we, instead of having 2 H's, we just have a propyl group. 1, 2, 3. And then we could further, and then we just reduce that. Okay. So that would work for B. Benzylamine. What's that? Benzene ring. Right? that's benzamide. Sorry. There we go. We could do this with both of them, yeah? We could use a benzonitrile.
Mhm.
We could use a benzamide, which is the thing I had earlier. In that case, either one would be good because we're putting that amine at the end of something. What about N-ethyl aniline? What does that structure look like? Aniline. Ooh. Aniline is a benzene ring, right?
Mhm.
With an amino group on it. So, N-ethyl if this is aniline, we have an N-ethyl, well, that just means that one of those hydrogens is an ethyl group. Okay. And how could we make that?
Chloride.
Yeah, exactly. So, we could do this. And then that's already in there. Oops. And then we reduce that down and end up with imine. And again, cyanide's not going to be a good candidate because that N is in between the two R groups. All right? That feel okay? All right. So, carbs, carbohydrate. Why is it called carbohydrate? Well, it turns out that that's just a carryover of a misunderstanding of the structure. So, when scientists first discovered what atoms were present in sugar, and what the molecular formula was, C6H12O6, they initially thought that it was a 6-carbon chain surrounded by water, which would make it a hydrate. You guys remember from Gen Chem, we talked about hydrates, or the nomenclature for hydrates. So magnesium sulfate dot 6-hydrate, right? That just means it has these water molecules attached to it. So they thought that this, they didn't realize this was the structure. They thought it was just 6 carbons linked together with a bunch of water molecules, because that would have technically worked. That would have been legit. It turns out it is not. But that's where the carbohydrate comes from. Carbon, hydrate, meaning that's the type of molecule it is. Okay? So we've come a long way since that thinking, but the name has still stuck with it. All right? I kind of talked a little bit about the importance of carbohydrates. This comes from the OpenStax, right? All living organisms have carbohydrates, whether they're humans, or pine trees, or bacteria. And not only do we use them to make energy, but we also have proteins that are glycosylated, meaning they have sugars attached to them. Receptors on the outside of our cells have sugars that give them their final properties. Okay, so protein synthesis, we'll talk about that a little bit later on next week. When a protein is made, it then goes to the Golgi and gets processed. Okay, and it's kind of like quality control. Okay, yep, you're a proper made protein. You're going to go here. Yep, you're going to go like an Amazon warehouse. You know, like, okay, here's the address. There you go. And then the final step is for them to get a certain sugar fingerprint attached to not all proteins, but a lot of them. Our red blood cells, our erythrocytes, are coated with sugars. Even if we aren't diabetic, right? We have sugars on our red blood cells, and that is what differentiates your blood type. Anybody know if you're A, AB, B, or O? Yeah. You're O?
I think I'm A positive.
A positive. You are.
I should
don't
know.
Luckily, they'll figure that out for you if you need to. But what makes you O, A, B, or AB is the types of sugars that are on those molecules, and so as well as some antibodies, some proteins. But that is why you can't just give anybody any type of blood, because that sugar profile and the antibodies associated with that will be recognized as foreign, and it will be attacked by the immune cells, and then the immune cells won't get the signal to calm down, they'll just start attacking your blood. Okay? And that's why O negative is a universal donor, because it doesn't have certain antibodies that would be recognized, and it's missing certain sugars. Okay? So carbohydrates are important not just from an energy perspective, but also from a physiological perspective as well. Finally, they make up our DNA, literally. So the nucleotides are attached to a sugar backbone, a phosphosugar backbone. And we'll touch on that toward the end of the lecture if we get to it. But so literally these sugars are a part of lots of different aspects other than just calories.
Is that part of how like the immune system recognizes like what cells like belong and what ones don't, or?
Not so much. That's more of a protein level. So what happens is every single cell in your body will take some of the proteins that it makes and just cut them up into a bunch of little pieces and then decorate itself with those pieces. And if those, and then your immune cells will go and they'll look at all the pieces. And it's just amazing the ability to detect a just slightly different shape, right? A point mutation, meaning one nucleotide can change the amino acid of that protein. One amino acid change can give it slightly a different conformation, right? And so the immune cells will read all of those fragments, and if they detect a fragment that is misfolded, that's a signal that there must be a mutation that's causing that to misfold, and they will attack that cell. And so that's why, even though we are frequently creating cancer cells in our bodies by random mutation and exposure to carcinogens and things like that, we don't always develop cancer from it, because our immune cells say, oh hey, that protein is mutated. Does that answer your question?
Yeah.
Yeah. So when you get an organ transplant, you have to be on all kinds of immunosuppressants, because the cells that make up that organ have proteins that come from an entirely different genome, right? And so you try to match it as best you can, but they will look at, your immune cells will start saying, okay, say you get a kidney transplant. They'll start looking at the cells in that kidney and saying, oh, I don't recognize these fragments. And then it will attack it. So yeah, it's kind of it's a really elegant way of our bodies to put these self-checking mechanisms in by just saying, okay, I need to make protein A for some response. Here we go. But we're going to we're going to make a little bit of it to just chop into pieces and decorate ourselves.
That's interesting.
It's kind of like waving the flag of being self or a compromised version. Questions other than that or in addition to that? All right, so how do we classify these? We classify them into two groups, aldose or ketose. And that's going to be based on what the carbonyl group is at one end. This is an open chain version of sugars. We're going to talk about how they most commonly exist as a ring, and we'll talk about how that ring forms. But for now, if you were to open this up, you have one end that either has an aldehyde group, hence aldose, or a ketone group here, right? A carbonyl in between two carbons. Ose is the suffix for sugar, okay? Sucrose, fructose, lactose, maltose, cellulose, etc. Okay, so we can take sugars and automatically put them into two different categories based on the function group at one end. All right. So let's determine what these are. We can also name them in terms of how many carbons they have. So this one here, is that an aldose or a ketose?
Aldose.
It's an aldose, and it is 1, 2, 3, 4, 5, 6. So we would call that an aldohexose. That tells us it has an aldehyde group, and it tells us that there are 6 carbons in that sugar. What about this one? What would we call this?
Aldopentose.
1, 2, 3, 4, 5. Aldopentose, exactly. How about this one?
Keto pentose.
Yeah, 1, 2, 3, 4, 5. Ketopentose. And then D, that's another aldo. 1, 2, 3, 4. So
Aldobutose.
aldobutose.
Sounds wrong.
And then what's this last one? Keto, right? And 1, 2, 3, 4, 5, 6. Ketohexose. And then of course these will have common names for them too. So a pentose will also in a ring form we'll call it a ribose. Other ones will have different names as well, but this is the easiest and more simplified way of just standardizing all of them. Okay. And it turns out that pretty much all naturally occurring sugars have a D conformation. And what does that mean? How do you know if something is D or L? Well, we look at the hydroxyl group on the chiral carbon furthest from the carbonyl. All right. And if it's on the right side, it's D. If it's on the left side, it's L. This last carbon, that's not chiral, right? Why isn't that chiral?
There's 2 hydrogens attached.
It has 2 hydrogens, right? So it doesn't have 4 distinct groups. So we never look at this one. We always start at the carbonyl end and go down to the very last chiral carbon and then say which side is that on? Is it on this side or this side? And as I mentioned, there are reactions that can make these sugars bigger or smaller. So they've reduced this glucose into a D-glyceraldehyde by trimming off those carbons in the middle. And it's not completely solidified why most natural carbohydrates are D, but it is believed that during evolutionary pressures, the enzymes that organisms had, for whatever reason, it's all about shape, right? So if you have an enzyme that can bind to a D-shaped sugar and process it more quickly, then fruits and sugars are going to respond to that, right? Because the whole reason a fruit is believed to exist is to be, for one, providing sugars to the seed, but then also it got kind of co-evolved into using a seed dispersal mechanism. If I have a really colorful, sweet berry, something will eat that, and the seeds will pass through it, and then they will scatter it around, right? And so if the things eating them just tend to gravitate more toward stuff they can process more quickly, then it drives these selective pressures to just have sugars that have that D instead of the L mirror image.
The plants evolved with the animals together.
Yep, exactly. So, also we make sugars. We can make sugars from our amino acids and stuff like that, from our lipids. And again, the enzymes that are used to break lipids down into sugars, if they can then process a D conformation more quickly, then it's just going to, over millions and millions of years, it's going to select for that formation. So that's a somewhat squishy definition, but that's the idea driving that, because that's what is observed. Most natural sugars just have that D.
So if you ate more like L sugars, would it be like harder to digest or for your body to break down, or can we pretty much like?
Not to my knowledge. I don't think so. I don't. It's not on a monosaccharide level. Now we'll get into polysaccharides and disaccharides, and depending on where certain hydroxyl groups are, it will create a disaccharide that our enzymes can't recognize and break down. Right? So cellulose, that is polysaccharide, and we can't break down cellulose. Right? Dogs can't break down cellulose. Anybody have a dog? Have they eaten grass before? What do? Come in and they throw it up. Cows can, because they have an enzyme that recognizes the structure of cellulose. And that structure literally is just based on where these methanol groups are. If they're all up, they alternate like this. So to your question though, if we were to switch this hydroxyl group over here, I'm not aware of any, indigestion consequences of that. Alright, so hopefully what's going on here stops soon. Let's practice. So, D or L? D. D or L? L.
D.
They're drawn up like that. That's easier.
So
Do we
pretty.
have that on the desk?
Yeah. is just going to be this question, okay? Yeah, so pretty straightforward how to do that. Now, let's talk about the different types. If we have, say, a pentose is going to have these 3 chiral centers, right? That's not chiral, that doesn't have 4, so that's not chiral. And so what that means is that there are essentially 8 possible enantiomers, depending on where we put these hydroxyl groups, right? So we can have them all on one side, or we could have, and then this would be D. L, of course, would be the mirror image and them all being on the left side. Right? Or we can then have these different iterations where we're putting hydroxyl on one side, 2 on the other side. Right? So with a pentose, with an aldopentose, we have 8 different possible conformations. As you might suspect, the more carbons we add, the more possibilities we have, right? So if we add 1 more carbon, if we go from a pentose to a hexose, now we've doubled it. We've got 16 possible enantiomers, right? Because, and you know, if you have a heptose, right, then it just keeps going. We're all on one side, 1 on one side, you know, alternate 2 and 2. Sugars don't get much larger than hexose. They can, but they don't. We're not going to have really some kind of decose sugar, really. Maybe you could make them, right? But in nature, they don't tend to exist that way very commonly. All right. So as I mentioned, most of these things exist in a ring, okay? Not in that open chain. That open chain is nice because it helps us see, you know, what the structure is. But in life, equilibrium greatly favors a ring. And it turns out that ring, a sugar ring, is, in some cases, a hemiacetal. So if you remember, we talked about acetals and hemiacetals before the midterm, so it's been several weeks now. But if we took an aldehyde and reacted it with an alcohol and then did an acid-catalyzed reaction, we can form what's called a hemiacetal, where we convert one of the OH groups essentially into an ether. And then if we were to tack on another R group onto this hydroxyl group, now we have an acetal, right? So this is hemi. If both of these are ORs, then it's acetal. If we were to take, let's say this, which is not necessarily a sugar, because it doesn't, a sugar is going to have either an aldehyde or a ketone on one end, but it also has hydroxyl groups throughout. But a generic form here, right, we would see that in its open chain, it's at equilibrium, it's not favored. Okay? It's going to drive much more toward closing this ring. And what happens is that this end circles around, right? And then usually there's a methyl group for with sugars. This is, again, this is generic, but with a sugar there's going to be another methyl group here. Because if we look back at those open chains, right? So this methyl group here, and it's going to do a little twist, and it's going to rotate. It's going to rotate so that the R group or the methyl group is out here, or the methanol group, and then we have this OH come into proximity with this carbonyl, breaks this bond, forms a bond here, and then the hydrogen jumps over. So it's nice, it's color-coded here. This bond, this double bond, goes to the oxygen, this hydrogen. That's kind of cool. I look like a little tracer there. Like, can you see? Is it just me?
Yeah.
It's not just me. Okay, good. All right. And then hops over there. Okay, and now we've closed that ring, and that's what sugars do. Okay, forming a hemiacetal. Now, if we end up with a ketone, then we're going to have an acetal, not a hemiacetal. So that's what we get with aldehyde sugars. Acetal is what we would get with ketone sugars. So let's practice. How would we close this ring, and what would that ring look like? One thing that is helpful is to keep track of the position of the carbons, okay? So we always number it starting at the carbonyl, 1, 2, 3, 4, okay? And this oxygen here, on this hydroxyl group, right? It's going to wrap around and bond to that double bond, right? And then this hydrogen is going to come around here and bond to that oxygen to form a hydroxyl group. So we're turning the carbonyl into a hydroxyl group, and we're turning the hydroxyl into an ether. So what would that ring look like? Well, we have an oxygen in there, right? This oxygen is going to be now in the ring. This one is in the ring, right? So then 1, 2, 3, 4, right? 1, 2, 3, 4. And then coming off that first carbon is our OH group. And then what's coming off of the third carbon?
Methyls.
Yeah. Everybody see that? It doesn't look like sorcery or anything, right? Okay. Because we're going to do some more practice here. Can I erase? Let's try closing these. And then we'll take a break. So again, 1, 2, 3. Now look here. This is, don't get hung up here. It goes beyond, right? But don't go 1, 2, 3, 4, 5. We're not doing longest chain. We're just counting the carbons in between the carbonyl and wherever that last hydroxyl group is, because that's what's going to come around. So these, even though the chain, the longest chain keeps going, these are just going to be methyl groups coming off of the ring. Same with this here. 1, 2, 3, 4, 5. We wouldn't go 6. That's not part of the ring. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4. Fine. So work with each other, work on your own, see if you can close those rings, and then we will go over them together.